नमस्कार। आप सभी लोग अपने अपने घरों पे सकुशल होंगे, इसी आशा के साथ हम बात करते हैं अपने टॉपिक द बायोटेक्नोलॉजी, इन विच वी हैव इन प्रीवियस लेक्चर वी हैव टॉक अबाउट द जनरल principles of biotechnology and the techniques of the genetic engineering as well as the construction of the first artificial recombinant DNA. Then we have to know that the different keys tools for the genetic engineering means how we can utilize the different tools for the such processes in the biotechnology. Means we can use the different tools, especially there are some tools that we can say that the restriction enzymes, cloning vector and competent host organism, or even instead of that we can say it may be restriction enzymes, polymerase enzymes, ligase, vector and host, right? Because these can be always helpful for us to do the different processes in this one. Only if we want to develop the recombinant DNA, in that particular conditions there are different enzymes which are very necessary for the making or preparation of the recombinant DNA. Especially we can say if we can talk about the restriction enzymes, they are also known as the molecular seizures. Especially we can utilize for the cutting of the DNA, right? So we can say especially the two enzymes from the Escherichia coli that were responsible for the restricting the growth of the bacteriophage were isolated in 1963, right? And one of them added in methyl group of the DNA and other will just other cut to DNA into a segment that the later on called the restriction endonuclease. Means in the starting in 1963, whenever the first two enzymes can be just used, or we can say utilized for the Escherichia coli for the during the growth of the bacteriophages over there, right? So in that condition they can be classified in such categories. Evenly if we can talk about the first restriction endonuclease enzyme, right, that the Hind second was the isolated, you must know the first endonuclease enzyme. Students, sometimes this 1 marks question can be asked over there. The first endonuclease enzyme that was Hind second, you can just name here that this is known as the Hind second, right? It can be generally isolated by the Smith, Wilcox and the Callery in 1968, right? They found in that in the always cut DNA molecules at a particular point by the recognizing of the specific sequences of the 6 base pairs known as the recognizing sequences. Means this Hind second can always utilize to cut at a particular place, particular sequences, that is known as the recognizing sequences in that particular condition. So it can be generally seen over there. Or we can say in 900 restriction enzymes isolated from the 230 strains of the bacteria. Even in that particular condition times we can say after the Hind second 900 restriction enzyme can be isolated from the 230 strains of the bacteria. In that conditions which can be recognized the different recognizing sequences, means different specific places of that particular conditions. So we can say generally these enzymes, these restricted enzymes are always belong to the nuclease class 2 enzymes. It's generally there are the two types which are found, which are known as the restriction endonuclease and the restriction exonuclease in that particular conditions, right? Especially if we can say that these enzymes, that exonuclease can always you must know, because sometimes the differences between these both enzymes can be asked over there. So the exonuclease, they can be always removed the nucleotide from the end of the DNA, that is either 5 prime or either 3 prime, right? Means one strand from the 2 prime. Generally if we can talk about especially about the this exonuclease, suppose this is a DNA sequence over there, and right, suppose this is the DNA sequences. So this exonuclease can be started from to cut the DNA from the 5 prime, right, or the 3 prime simply. So these such type of enzymes is known as the exonuclease in that particular condition. On the other hand, the endonuclease enzyme, right, they are can be always, these enzymes can also a specific structure, but they can show that they can always cut such things in a particular place, right? We can say, suppose this is an enzyme, this is an DNA. Now after that, right, they can always utilize to cut at a particular place, right, from any place like this. So that's why it is a molecular precision. But it be not started from any end. It can be start or it can be cut from the middle part of the DNA. So in that particular condition, we can say the endonuclease can be always cut at a specific position within the DNA, and these specific sequences are recognized sequences. Especially, we can say in that particular conditions, each restriction endonuclease base means especially the each restriction endonuclease recognize the specific palindromic nucleotide sequence in the DNA. I will just going to explain you after few time that the palindromic syndrome, right? What about this palindromic? Palindrome क्या होता है अभी बताता हूँ थोड़ी देर में बेटा. But first of all, you know, especially the palindrome क्या है? The palindrome, we can say it is the sequence of the DNA base pair that reads same on the two strands when orientation of reading is kept same. Means generally अगर हम बात करते हैं palindromic सीक्वेंस की, तो पैलिंड्रोमिक सिंड्रोम सीक्वेंस क्या होगा कि वो हमेशा बिल्कुल सेम टू सेम होगा दोनों तरफ से, यानी कि 5 to 3 एंड 3 to 5 वो हमेशा क्या होगा, सेम टू सेम होगा. यानी कि सपोज वी कैन ड्रॉ अ स्ट्रक्चर लाइक दिस, राइट. सो स्पेशली इन दैट पर्टिकुलर कंडीशन यू मस्ट फॉर्म दैट in this sequences, like you can say G A T T and so on C. So on the other hand you can say G A T T C. अगर आप ध्यान से देखेंगे बेटा, तो अगर हम मान लो ये 5 प्राइम है, ये 3 प्राइम है, और यहाँ पे 5 प्राइम और ये 3 प्राइम है, तो the sequence of the base pairs is always same. Means sometimes you must know it. Means especially we can say whenever we can start to read this they should be always similar in the direction 5 prime to 3 prime as well as 3 prime to 5 prime. Means they should be not differ from each other. So such sequences we can say the palindromic syndrome. As we could simply, आप अगर हम जनरल लाइफ की बात करते हैं, अगर हम लिखते हैं मलयालम, जस्ट स्पेल द वर्ड मलयालम, राइट. एंड मलयालम को अगर हम सीधे बोलते हैं मलयालम, अगर हम उसको उल्टा भी पढ़ेंगे तो भी क्या होगा, मलयालम ही लिख के आएगा. सो दैट स्ट्रक्चर एक जनरल, आपने बचपन में एक कहानी सुनी होगी, एक वर्ड सुना होगा कि जैसे हम देखते हैं कि जहाज, डालडा, ऐसा वर्ड जो उल्टी तरफ से और सीधी तरफ से सेम टू सेम आए. तो इसी सीक्वेंसेस को अगर हम बात करते हैं इन बायोटेक्नोलॉजी, तो दैट इज नोन एज पैलिंड्रोम. स्पेशली द डीएनए सीक्वेंसेस और बेस पेयर दैट रीड सेम ऑन द टू स्ट्रैंड्स whenever the orientation of the reading is done. जब डीएनए के दोनों सीक्वेंस पे उसको रीड करेंगे, चाहे वो 5 प्राइम टू 3 प्राइम हो, चाहे 3 प्राइम टू 5 प्राइम हो, दे कैन बी ऑलवेज सेम, right. So those sequences can be cut with the help of the endonuclease enzyme. So it is very important for point of view of this one. I think you will understand about this especially one. Evenly there are some other things are also necessary for that one, especially if we can talk about this one, that restriction enzymes can inspect the DNA sequences to find the specific region, as I told you. Especially they can be have a site to bind and cut, especially each of the two strands that the double helix at their sugar phosphate backbone. Evenly we can say these restriction enzyme can recognize the palindromic nucleotide sequences in the DNA, as you can just see in the following situations in the over here, right. You can say it is given here. You can just take the another one also, but it is just given for real examples only. After that we can say the another thing, that how this restriction enzymes can show their action. Means how this restriction enzyme can show their action in that particular conditions. Right. For that we have to know especially the mechanism of the action of the restriction enzyme. First of all, we can say the restriction enzymes cut the strands of the DNA at a little away from the center of the palindrome sites. You always think about this one. Especially the restriction enzyme can cut the strand of DNA a little away from the center of palindrome sides, but between the same two bases on the opposite sides. मीन जनरली ये सेंटर से थोड़ा साइड में हटके कट करेंगे, लेकिन वो दोनों साइड से अपोजिट में क्या होना चाहिए, सेम टू सेम होने चाहिए, इसका ध्यान रखना पड़ता है. साथ ही वी कैन से दैट द especially these restriction enzyme can be leave the single standard point at the ends, especially they can be leave over there. Evenly we can say the overhanging strand called the sticky ends, especially the we can say the which can be attached to the helix structure, or we simply directly you can say the overhanging strands, they are known as sticky ends on the each strands as given generally shown in the diagrams. You can just see in this diagrams that endonuclease enzyme can introduce over there, it can be cut in that particular place and in that particular place over there. So if we get the such structures, one structures that can be over this, these are the sticky ends and these are the sticky ends, these are the part of this one, right? So they can be always provided such sticky ends over there or hanging over there. Evenly अब sometimes we can say why they are known as this sticky ends, आखिर इनको sticky ends क्यों कहा जाता है, राइट? तो इसके पीछे भी एक रीजन है, बिकॉज जनरली दे कैन हैव टू फॉर्म हाइड्रोजन बॉन्ड विद अदर कॉम्प्लीमेंट्री कट काउंटर पार्ट्स. मीन्स जनरली ये जो एंड्स आपके सामने दिखाई दे रहे हैं, दे कैन कैपेबल टू मेक द हाइड्रोजन बॉन्ड्स, अपने ही जैसे दूसरे कॉम्प्लीमेंट्री के साथ अपना हाइड्रोजन बॉन्डिंग करते हैं, इसलिए these ends are known as the sticky ends in that particular condition. Even in the given diagram you can see different types of the restriction enzymes which can be cut in different conditions. Especially there is one more thing, that sticky ends hydrogen bonds which can be complementary cut counterparts with the help of a DNA ligase. Especially we all have to know that these stickiness means generally these sticky ends can be capable to attach to other parts only with the help of the DNA ligase, because this can be always helpful to attaching or the sticking of the other complementary parts, or where they can be going to be counterparts over, right? So they can be always helpful for the attachment over there, as you can just see especially in this diagram over there. So this is a very important one, right? So many times this diagram is asked in the examination, but before that we have to complete it. We have to know some more things about this restriction endonuclease enzymes. Especially the restriction endonuclease enzymes, they are used in genetic engineering to form the recombinant molecule of DNA, means to formation of the recombinant DNA, which can are composed by the DNA from the different sources of the genome. We can be always get the recombinant DNA from the different places. Evenly we can say the sticky ends are complementary to the each other when they cut the same restriction enzymes. Therefore we can join together end to end using DNA ligase. But students, you must know the special things, right, about this one, that suppose we are going to use of this restriction enzyme to for the formation of the recombinant DNA. In that particular condition, you must know that first of all how this restriction enzyme can act over there. Even you know the mechanism of restriction enzymes, but you must know one special thing, that the vector DNA and the foreign DNA. Vector DNA, the DNA which can be utilizing us where we are taking it, and foreign DNA where we have to just adding it, right? In that particular conditions we have to know that how they can be utilized over there. For that special condition, we for the cutting of this both vector DNA and the foreign DNA, we have to always utilize the same restriction enzymes. Right? It is very necessary. It is very important point that without using the same restriction enzymes, they can be just cut at any from any places. Your such process cannot be completed over there. Means in that particular condition, you have to needed the special and the same kinds of the restriction enzymes, as you can just see in the diagrams. In that this particular diagrams, you can just see, especially this is the vector DNA and this is the foreign DNA. In that particular conditions, here the foreign DNA and here the vector DNA, the E. coli, means especially here we are using in that particular condition, in this particular condition, and this, and over here the same kinds of the endonuclease enzyme can be utilized over there. So the same kinds of sticky ends and they can be cutted in vector as well as in the foreign DNA. Means we will get the proper structures of the counter or complementary parts in that particular conditions. So they can capable to ends with the help of the ligase enzymes. As you can just see that after the action of this here, the restriction enzymes, they can cut it and make the sticky ends in both the strands, and after that we can utilize the ligase, DNA ligase, which can be able to join them with the help of hydrogen bonding with the help of that for vector DNA and the foreign DNA into each other. This is the general process for the attachment of these restriction enzymes. But there is one more thing, that how these restriction enzymes can be given their name in the nomenclature of the restriction enzymes. Generally in this book you all have just seen this E. coli, right, or Pestle, smell, and the hell and the apple. There are different enzymes which can be used over there. So how they are given that such type of name, especially if it known about their nomenclature. So first of all, we know the first letter which can be always derived from the genus name and next two letters from the species name of the prokaryotic cells from which the enzymes are extracted. Simply, as I have told you in the 11th class, in the same way, the first letter of which can be belongs to the genus as well as the second two letters are always belong to the species from where or from the which prokaryotic cell they are can be obtained or extracted from over there. Evenly, the Roman numbers after name also show the order in which the enzymes were isolated from the bacterial strain. Mean when हम जिससे कहते हैं हिंदी सेकंड, राइट, जो रोमन नंबर्स की लाइन हम खींचते हैं, रोमन नंबर्स लिखते हैं, दे कैन बी ऑलवेज हेल्पफुल टू शोज द ऑर्डर इन विच द एंजाइम्स वर आइसोलेटेड. किस ऑर्डर पे उसको आइसोलेट किया गया है इसको भी शो करते हैं. स्पेशली एज वी ऑल नो द इसेर्सिया कोराई, इसेर्सिया E-coli first suppose, right, and that is known as Escherichia coli RY13 and E-coli second comes from the E-coli 245. Especially we have to know, जैसे हम बात करते हैं, मान लीजिए कि Escherichia coli first, तो Escherichia coli first generally it can be obtained from the Escherichia coli RY13 bacteria. Evenly the Escherichia coli second which can be come from the Escherichia coli R245, means that different just excreted from the different places over there. So this is the general ways through which we can capable to name all such kinds of structures over there. Evenly one more thing which is given in some books but it is not given in some books sometimes. So that is mean how this recombinant DNA utilized even in that particular condition. So you can just see over there we can utilize this is the foreign DNA, right? This is a foreign DNA and this is the vector DNA. So whenever the restriction enzyme can introduce in both of them, they can be capable to foreign DNA can cuts like this parts and it can be also loses the such parts. right? Means they can be always cut in a particular sequence, in a specific sequences, palindromic sequences, and after that we can attach the foreign DNA and to the vector DNA with their sticky ends with the help of the enzyme that is known as DNA ligase, right? And when we will get the such structures, they can be again, we will get the recombinant DNA, and that should be just introduced into the a capable host. As you can see, especially we are taking the Escherichia coli. So this recombinant DNA can be utilized to just introduce in the this host. This process for the hosting of in that particular condition, that is not transformation, and when that cell can start to develop and grow and divided, so we will get the large amount of the such recombinant DNA, and we can capable to utilize, take them from there, and we can utilize in our biotechnology, in genetic engineering, in different ways. So this is the mechanism of the biotechnology, students. So if you have not understand, you can just contact me, or you can just give me your calls, though I will again try to clarify about all these topics. So students, you must complete your notes with the diagrams also, because there is so many times this diagram is asked in the examination, as well as this diagram is also many times asked over there. So you must just pay attention over all these things, especially the types of the restriction enzymes, why it is known as the molecular seizures, where it can be utilized, and how they can be discovered over, how they can be given names over there, and so on. So, and how they can, what is the palindromes. You all must just memorize all these things. They are very important in the point of view of this such things. So this way this topic is completed. So in next videos we will talk about the next, that is called the separation of isolation of DNA fragments. How we can capable to separate the DNA fragments in different ways. So complete your notes once again. Stay in homes and safe. Thank you.